Live cell imaging is particularly useful when studying the dynamics of organelle trafficking. This video will highlight a procedure used to image dense core vesicles in cultured hippocampal neurons. Transfection complexes are first obtained by mixing DNA, MEM medium and the lipofectamine transfection reagent. Since hippocampal neurons are cultured on inverted cover slips facing a glial monolayer, these cover slips must be turned over in order to expose them to the transfection reagent. After allowing ample time for expression of GFP-tagged dense core vesicle proteins, an imaging chamber is constructed and imaging of dense core vesicles in cultured neurons can be performed using wide-field fluorescence microscopy. This protocol is flexible and can be adapted to image other organelles such as mitochondria, endosomes and peroxisomes.
Hi, I'm Michael Silverman and welcome to my laboratory for cellular neuroscience at Simon Fraser University. We're interested in how organelles move in nerve cells. To approach this question, we label membrane-bound organelles, specifically dense core vesicles, in cultured hippocampal neurons. And to show you how we do this, I'm going to turn it over to David.
Hi, I'm David from the Silverman lab. Today I'll show you a procedure for live cell imaging of dense core vesicles in cultured neurons using wide-field fluorescence microscopy. Let's get started.
For most experiments, we use dishes of rat hippocampal neurons that have been cultured at a cell density of 250,000 cells per 6 cm dish. For each transfection, label two 1.5 milliliter tubes, one for the plasmid DNA and the other for the transfection reagent. In a laminar flow hood, combine 1 microgram of plasmid DNA with 100 microliters of MEM in a single tube at room temperature. Be sure to use MEM that does not contain any supplements.